We use bacteria as our model when we talk about transcription because transcription in bacteria is somewhat simpler than transcription in eukaryotes. There are a lot of things that overlap, but in general, physiological processes in bacteria are simpler than the same processes in eukaryotes because bacteria are more simple organisms. So what I'd like to do now is move on to talk about some of the major differences between transcription in eukaryotes and transcription in bacteria. The first thing I'd like to talk about is this point right here, which states that there are proteins called basal transcription factors, or BTFs, in eukaryotes that help initiate transcription by bringing the RNA polymerase enzyme into its appropriate promoter region. Now, this sounds a lot like what sigma does, and indeed, the basal transcription factors that you find in eukaryotic organisms essentially are analogous to sigma. They are regulatory. They help get the core of the RNA polymerase enzyme to the correct location on the DNA where transcription should begin. One of the differences between sigma in bacteria and BTFs in eukaryotes is that BTFs can come in and bind to and interact with promoters on the DNA without the rest of the RNA polymerase enzyme. In other words, they can come in and bind without having the core alongside them. In bacteria, in addition, a single sigma protein binds to the promoter to initiate transcription, whereas in eukaryotes, there are many BTFs that are required to do this. So the initiation of transcription is one place where we see much more complexity in eukaryotes than in bacteria. The next thing to make note of is that, unlike bacteria, eukaryotic organisms actually have three different types of RNA polymerase in their cells. There's RNA polymerase 1, RNA polymerase 2, and RNA polymerase 3. These individual types of RNA polymerase have a different role in terms of what kind of RNA they produce. In other words, if you look at RNA polymerase 2, this is the enzyme that's analogous to RNA polymerase in bacteria in that it produces mRNA, or messenger RNA. This is the type of RNA that encodes for protein production. RNA polymerase 1, in comparison, codes for the genes that help produce rRNA, and RNA polymerase 3, down here, helps produce some additional different molecules, including what are called snRNPs, which we'll talk about in a little while, as well as tRNAs. So there are some pretty significant differences here in that bacteria only have one type of RNA polymerase, which corresponds with RNA polymerase 2 in eukaryotes. Another thing to make note of when we make comparisons between bacterial organisms and eukaryotic organisms is the promoter regions. The promoter regions in eukaryotes are much more diverse and also more complex. Remember, we said in bacteria we typically encounter a -10 box and a -35 box. Well, RNA polymerase can recognize a particular promoter that has a sequence of TA-TA, known as the TATA box or TATA box, and this is 30 base pairs upstream, or -30 from the +1 transcription start site. Now, this is a common promoter region that's recognized by RNA polymerase 2, but it's certainly not the only promoter region that's recognized by RNA polymerase 2. So we have many promoter regions. We see a great diversity, in fact, in promoter regions in eukaryotic organisms. Another thing to notice is that in eukaryotic transcription, the RNA that's produced is not a mature RNA. In other words, some processing has to be done to the molecule before it can leave the nucleus and go on to be translated. The first part of that processing involves removing pieces of the RNA molecule that are not necessary for translation. It turns out that eukaryotic genes don't consist of one long continuous piece of DNA. That might seem a little counterintuitive to you, but genes are often divided into pieces in eukaryotes. So the RNA that is made from those genes also can be thought of as being in pieces. Pieces that are coding pieces that are necessary to make a protein, and pieces that need to be removed. And these pieces are referred to as exons and introns. The exon part of the RNA molecule is the portion that is going to be used to make proteins. It's going to be part of the final mRNA, or the mature mRNA. In comparison, introns are intervening sequences, sequences that are not going to be part of the final mRNA and need to be removed. So in this slide, we have a couple of different images that all represent the same thing. Over here, you can actually see a micrograph of a Dna Rna hybrid molecule. It's hard to make out a lot of detail here, but we have the DNA shown in red in this picture, and the RNA shown in yellow. Okay, so this is the interaction that we see during transcription when RNA is made from DNA. Over here is where I want you to direct your attention next. Over here you can see up top this red and pink strand. This represents an immature molecule of mRNA. So this is what's produced during transcription. It's a complementary copy of the template strand of DNA. Now look down at the bottom in yellow. This is the mature mRNA that will be used as the mature transcript, or the piece of information that will go out into the cytoplasm of the cell and be used as the basis of translation. The only difference between this red and pink molecule and this yellow molecule is that pieces have been removed, pieces in pink, and the remaining red regions have been joined together to help produce this yellow molecule. So this process of taking out regions of an immature RNA, this process is known as splicing. And what has to happen is that the RNA polymerase II molecule has to first create what we call the immature or the primary RNA transcript. And this will contain both exons and introns. Exons that have important information for making a protein, and introns that need to be removed. As transcription proceeds in eukaryotes, the introns are going to be removed through splicing. So again, down here you can see what represents the DNA. Here's the promoter where the basal transcription factors would bind, and the core of the enzyme would come in and begin to transcribe this material. Here's the mRNA that's produced. It contains both exons in the darker yellow and introns in the lighter yellow. The cell needs to remove those or splice those out and then join the remaining dark yellow pieces together. So this is achieved with a molecule that is formed out of a group of proteins and small RNAs that's called small nuclear ribonucleoproteins. Small nuclear ribonucleoproteins, or what we shorthand as SNRNPs, which we call SNRPs. And for our purposes in this course, you can refer to them as SNRPs because everyone else does. Now, SNRPs can be thought of as sort of editors of the transcript or the immature form of the RNA molecule. The SNRPs are going to come in and they're going to bind at the boundary of exons and introns. This generates a complex that's referred to as a spliceosome. A spliceosome, and this is a term I want us to be familiar with. A spliceosome is basically a complex of a group of SNRPs all bound to the RNA molecule. The spliceosome contains up to 145 proteins and RNAs in humans. So this is a massive, massive molecule. Please note that here on the slide where it says DNA, that should actually say RNA. We're talking about the RNA molecule here. Now, the spliceosome is going to be binding again at the boundary between an intron and an exon. So the intron will be drawn up into a loop, and that loop is going to have a single A base or an adenine base right at its boundary. And I'll show you what I mean in this next slide. So in yellow up at the top, we can see two exons with an intron in between them. These purple circular molecules represent SNRPs. All of the SNRPs are going to come in and they're going to help remove this intron section. You'll notice that there's a capital A here that represents the single adenine that's going to be at the boundary between this intron and this exon. So the SNRPs come in and they bind at the boundary between the exons and the intron, here and here. And that binding causes the intron to loop up. Okay? So essentially what the SNRP is doing, the spliceosome I should say, is bringing the exons closer together and making the intron loop up. That adenine at the end of the intron, at the boundary between the intron and the exon, is actually going to participate in a reaction where the exon is bound to the exon and the intron is removed. So the adenine helps participate along with the spliceosomes, or spliceosome, in bringing the two exon boundaries together and building a phosphodiester bond between them, and actually cleaving or removing the intron. Now, a lot of students wonder what happens to these introns. What's the cell going to do with these introns? And the answer is that the cell's just going to recycle them. Those introns, those strands of RNA, will simply be recycled into individual ribonucleotides and they'll be reused. Now, within the spliceosome, we have to have some catalytic activity because we're building, ultimately, a covalent bond between the two exons. So the cutting and joining of these exons is catalyzed by the spliceosome, by an enzymatic or catalytic region in the spliceosome, and that region is where the RNA is located. Okay, remember, the spliceosome contains SNRPs, and the SNRPs are composed of both proteins and RNAs. And these are very special RNAs called ribozymes. They're not enzymes. Okay, by definition, an enzyme is a protein. These RNAs are called ribozymes. They are catalytic RNA. So in this slide, we can see the final pieces of the spliceosome mechanism going on. Here we can see this purple molecule. These are the SNRPs all joined together into a spliceosome. We can see this exon, and we can see the looped intron with that adenine exposed at one end. The spliceosome is actually going to cleave off this exon, cleave off this exon, and then join them together with the ribozyme, with the catalytic RNA, joining a covalent phosphodiester, or building a covalent phosphodiester bond in the center of the molecule. Here's the intron that's left behind, and this will be recycled and reused by the cell. So we've gone from this immature RNA, which has introns and exons in it, to a more mature RNA, which only contains exons. All of the introns have been removed. This is still not the point of complete maturation of the RNA molecule, however. There is one final thing that needs to be done, and that's capping and tailing this molecule. So we're going to add a structure to one end of the RNA called a cap, and we're going to add a structure to the other end of the RNA called a tail. The cap structure is a structure that actually helps protect the mRNA from degradation. This mRNA, as you remember from earlier lecture material, is never going to assume a full tertiary shape, so it is definitely susceptible to degradation. The cell only needs this mRNA to survive long enough to be transcribed, but it does need to survive that long. And that's one of the things that the cap does, is it protects it so it can finish its work. The tail on the other end of the molecule is going to help regulate translation. Remember, translation is the process where this mRNA molecule is used to make a protein. So, in terms of the cap, the 5' end of this mRNA molecule is going to have a cap added to it in the form of a molecule called 7-methylguanylate, or M7G, in addition to 3 phosphate groups. Okay, so we're going to place at the 5' end M7G and 3 phosphate groups, and this is going to function as the cap. At the other end, at the 3' end, another enzyme will come along and add the tail, which consists of a long chain, about 100 to 250 or so adenines. This tail is often referred to as a poly-A tail. And again, its job is going to help to regulate translation. So we finally have our mature transcript, and that's what you can see in this slide. We've got our exons all joined together. Then we've got a couple of regions at each end that are referred to as untranslated regions. These are regulatory regions. Then we've got a cap up here, the M7G molecule and 3 phosphate groups. And at the other end we have a poly-A tail. So this mRNA is ready to go into the cytoplasm and be translated into protein. This last slide for us today is a table that comes directly out of your textbook. And I put it in here because it helps us make the comparison between transcription in bacteria and transcription in eukaryotes. So on the very left-hand side of the table, they provide different aspects of transcription, things like the RNA polymerase enzyme, the promoter structure, the proteins involved, and any processing steps that occur. And then they compare in this column what's going on in bacteria to this column what's going on in eukaryotes. For example, in terms of the RNA polymerase enzyme, in bacteria there's only one, and it's called Rna polymerase. But in eukaryotes there are 3. And each of these 3 different kinds of RNA polymerase helps produce 3 different kinds of RNA. mRNA is produced by RNA polymerase 2. tRNA is produced by RNA polymerase 3. And rRNA is produced by RNA polymerase 1. So that's a pretty big difference between these 2 groups of organisms. And as you move down the table, you can see comparisons between the other aspects of transcription. Now, when we get together again in the classroom, our next discussion will be what's going to happen to our mature RNA transcript as it comes out of the nucleus and moves into the cytoplasm to undergo this process that we call translation.